All right, good morning, everyone. So the second lecture this morning, I will be talking about how to read and analyze high throughput data. So visualization and interpretation of high throughput genomic data. So my name is Chongji Zhang. I'm associate professor in the Department of Genome Sciences, and I also work as the director for computational genomics in the Cancer Center. So the QR code goes to my lab if you're interested in what my lab is doing. So my lab is a computational biology lab. We develop computational methods for better analyzing high throughput omics data and to use the computational and data science approaches to study epigenetics and transcription regulation in chromatin. So we use data approaches. So all I'm going to talk about today is about big data that probably none of us has an idea of what this is about. But in today's lecture, I'm not going to discuss how to analyze big data, how to write code, or how to use software to analyze big data. Instead, regardless of whether you will be involving in processing or do this type of a big data analysis, when you read literature, you read papers, you will see a lot of data that presented, high throughput data presented in the papers, in the literature, in various different ways. So the goal of this lecture is for you to understand these type of data presentation. So a few learning objectives for today's lecture includes understand different types of plots commonly used for presenting these different type of high throughput omics data. And most importantly, when you see a figure in a paper, you want to understand the essential elements in these data, in the what information that these plots gives, and what type of data and what message you can get from different type of data. And also, I will share with you some tips for data presentation. So when everyone of us are doing research, we want to present our data in a graphic way or in a certain way that people can directly understand, instead of stacking an Excel table of just a huge amount of numbers, right? So graphic presentation of data is very important. And if you think about the fundamental message that you want your readers or reviewers to understand what type of information you want to present the data. So there are actually not many different types of messages. So I just give you a few examples. So if you observe something, if you see something, you'll see a signal, and you want to present it as compared to some background or control. So that kind of message is that's yes or no. You see something, you don't see something. Something exists, something doesn't exist. So yes or no kind of comparison. And the second type of presentation is you want to show two quantities that you want to show that one is, you want to do a comparison. One is bigger than the other, right? Once you show like you run the gel to do a Western blot or something, you see the band or you don't see the band. That's kind of a yes or no. But sometimes you see a very strong, dense band or compared to a weak band. So that's a big quantitative comparison, yes or no. And if you see the some measurements, say the weight or size of a mouse, it's a big or small comparison. And if there are more numbers, not a simple comparison between the two, you want to see whether the multiple numbers have a certain type of a correlation, right? Especially if the data becomes two-dimensional. You want to see whether the numbers have some correlation or more complicated type of patterns. So keep in mind this type of information in mind. That's the fundamental messages you want to get from all of different type of plots. So just give you some examples that you frequently see, especially for omics data presentation. You can see these type of plots. Can someone tell what this type of plot is from? We're using what type of tool that you can generate or visualize this type of plot? Anyone? Yes, you're talking about IGV. It's called Integrative Genome Viewer. So it's kind of a genome browser. It's type of tool to visualize different types of genomic data. And you can see different tracks and a lot of spikes or peaks. And some other regions looks like background or noises. And what do they mean? And if you zoom in these type of plots, you can see these like mountains and valleys and peaks, all of those signals. What do they mean? So this is the most typical way to directly visualize high throughput sequencing data from a genome browser. So we can see all of these type of plots. We're looking at certain regions in the genome. So over 30 years ago, there was the Human Genome Project that sequenced the whole genome sequences of human. And now, over the past 20 years or so, people update the genome version to make a most accurate ever coordinate system to record all of the sequences in the genome and create this coordinate system. It's just the chromosomal coordinates, include which chromosome it is and the some coordinates. So that's in this way we know where in the genome we're looking at, and we can see the certain scale. And this is just right at this coordinate system. We look at some signals. We'll go into more details about this. So before doing that, I'll briefly go through how the high throughput short read sequencing works so that we can understand how these data presentation or how the data come from. Some of you were with me in Group E when we discussed about Hi-C and the chromatin capture technologies, where I briefly mentioned for Hi-C technologies, the high throughput sequencing-based version. So most of the currently most commonly used sequencing technology, high throughput sequencing using the Illumina technology, which was invented in the year of around 2005, with the first commercially available machine available in 2006. And the idea, instead of the first generation sequencing, where it's very expensive and slow, so the high throughput sequencing can generate millions of short sequence reads. And it has been applied to all different types of sequencing-based biochemical assays. You probably heard some of them, like RNA-seq, ChIP-seq, ATAC-seq, or Hi-C, or more recent years when people do cut and run, cut and tag. And you've heard about the increasing amount of work working on single cell sequencing. Then most of them are based on these Illumina-based short read sequences. So the idea is that we got a large amount of DNA fragments. And what the Illumina machine does is that they can sequence the end of the input DNA sequences, either single end, in which they can randomly pick one end to get the sequence, or paired end, in which they can sequence both ends of that DNA fragments at the same time. And the sequence read means this blue or red part, sequence read, is just the end of sequence, and usually it's equal length. Nowadays, most sequences is about 150 base pairs on both ends, regardless of what the entire length of that DNA fragments. So keep in mind that all of this short read sequencing, they don't guarantee to give you the sequence of the whole DNA fragments, but they can only give you the end, one end or both ends of the DNA fragments. And now, so we started with millions of these short sequences. And what we do is that we align each and every of these short sequences to the reference genome, because we know the entire reference human genome, and we map them and to just pile them up into different places in the genome. So this is just like we map the short reads in certain regions in the genome. And we can see this is an example of certain regions in the genome. There are a lot of reads stacked together, but still, they are not easy to visualize. Now, how to visualize this type of data in a more straightforward way? What people do is that they generate these signal tracks information. So how these tracks are generated? So at the beginning, we have the short reads, short sequence reads, and if they are paired, we directly know the entire DNA sequence, right? From the mapped position of the two ends, we reconstruct the whole DNA sequence, DNA fragments. And if it's a single end, we usually estimate the average length of the whole DNA fragments and extend from the end read sequence to the entire DNA fragments. And we simply, based on the location of each of these sequence reads mapped to the genome, we just map the entire, just put the entire DNA fragments in the genome and pile them up. And after pile them up, we can generate these type of visualization. Any question so far? Yes.
Previous slide, why are the forward and reverse reads kind of localized? I would imagine that they overlap somewhat based on the base pair regions.
Yeah, that's a good question. So this type of picture, this type of pattern just means that there are a lot of DNA sequences, right? So some sequences, you have their 5' and reads mapped to this position. Some of them, you have their right parents. So this is from a single end sequence. So we don't know, we don't guarantee that we have every sequence read that have their corresponding at the end. So this is just usually people what we observe. It can be very messy, can be noisy, but we can see there are both the sequences can be mapped to either way. Yeah. So once we generate these type of tracks, we can see the different type of data format to represent these type of data. So one type of data format is called backgraph. So you can easily understand that directly give you the chromosomal coordinates, which chromosome it is, and what is the start position and what is end position. Usually they generate this means different bins. So from this particular example, this means a bin of 50 base pairs, so 200 to 250. And in this bin, there are 5 reads out of, or the signal strength is 5. So we can see it's just each different bin will have a different number. And then if we have a lot number of rows in this data, we can plot this in the genome browser. And then the easier format that saves some space, storage space, is called a wiggle format. So because we know for bad graph, you have to define every row, every line, you have to define which chromosome it is. But usually the same chromosome, the data from the same chromosome they occur together, so we can just label this which chromosome it is and what is the span, or in other words, what's the bin size, so that you don't have to record the start and end in every line. So in this case, it saves nearly half of the storage space to store this type of data. And another commonly used data, so if you download some publicly available omics sequencing data, you can see the commonly used data format called BigWig. BigWig is a binary coded format of wiggle files that you can directly download and load on the genome browser to look at this. So as I mentioned, what are the essential elements in these genome browser tracks? So first, we want to know the chromosomal locations. That every plot, they should label that which chromosome it is and what are the locations, the coordinates they should label there. Otherwise, you have no idea where this data is coming from. And the track label. Track label means that what type of data they are showing. So every track represents one data sample, one sample of sequencing data. And the scale, always the scale is very important. And the x-axis, what is the resolution of the data, what is the range of the genome that we are looking at. So for this, in this example, this whole window is about 50 kilobases. That is usually bigger than the average size of a gene. So, and we can see there are a lot of annotated genes labeled here. So this whole region cover multiple genes. And the y-axis means the height or the number of sequence reads with or without normalization, showing from each of these different data sets. So once there's a peak, there's a lot of signals or sequence reads piled up together, we know that there is some signal. So this is how to visualize or how to understand this tracks on the genome browser. Another type of data visualization for these omics data is that sometimes we want to look at such peak signals from many different places in the genome. So for example, there are over 20,000 genes in the human genome. If we want to see whether some epigenomic markers like histone modifications or transcription factor bindings, whether they occur in transcription start region or promoter region of genome, we want to look at tens of thousands of gene promoters together. So simply by using genome browser, it's probably very difficult. You need to look at every gene at a time. And you cannot put 20,000 snapshots of genome browser in the same time. So another way of visualizing this is to stack a lot of browser snapshots together to generate this kind of a heat map, or it looks like the water ripple. So when we see this type of heat map, what does it mean? So the idea is to stack multiple or many different regions. So for each region actually comes from a snapshot from the genome browser. And we can convert, so in the genome browser snapshot, the signal strength is represented by the height of the track of the signal. And instead of looking at the height, we convert this into one line, the one-dimensional one line, but use a color scale. That's what is called a heat map. Use a color to represent the signal strength. So if in the genome browser it's higher, we give this a more dense color. If it's lower, we use a different color. So in this way, we have one line, one row with different colors. And now if we have hundreds of different rows, we can align them and stack them together to generate these type of heat map. So if after the align, if we see some pattern in the heat map, we can observe that some type of signal are commonly occurred or recursively occurred in many different regions in the genome. So the essential elements in such, yes?
When you decided you want to do the paired reads, and when you decided you want to do the single reads?
Oh yeah, so whether there's a pair end or single end, that actually depends on the sequencing experiment. So you want to do this generate pair ends, so the sequencer will directly give you all the sequences that includes the information, the sequences from both ends. But if you don't set the sequencing experiments as pair end, they can give you the information from single end. It's not from the data, but well, in the meantime, the parent data can always be treated as single end. You just treat them separately, but the single end data, sequence data cannot be treated as parent because you don't have the information from the other.
Well, when do you decide what type of data you want to send in for sequence?
So that you have to make your decision to tell the when you send the sequencing, you need to decide whether you want to do the single end or parent, depending on which specific type of experiment. So usually for RNA-seq, either single end or pair end should work. Because you want well, if you're only interested in like the coding gene expression level, single end should be okay. But nowadays, more people tend to use pair end because pair end can give you more information, like alternative splicing. So because for alternative splicing, you're not simply going to map a sequence reads in certain places, but a single end can give you more information about the whole DNA fragments and give you more information if alternative splicing occurs. So just an example. For ChIP-seq, for example, you want to detect the DNA fragments associated with a certain histone modifications or transcription factor bindings. Usually pair end works better because you get the whole DNA fragments. Yeah. Yeah. So about this heat map. So first, the heat map represents three-dimensional data, right? The x-axis, y-axis, and the heat level, heat density, the color density. So whenever we see a heat map, we want to understand what are these three dimensions and what each of the dimension means. So x-axis tells what locations are each row. And usually because these locations have to be lined up with some anchor. So what is the anchor? So for example, this is looking at some transcription star size of hundreds of thousands of genes. They are aligned according to their transcription star size coordinates. So that's the anchor. And what is the range? So this plot they are looking at minus 1 kb to plus 1 kb from the transcription star size. And y-axis, what are each different rows? So how many genes or how many genes TSS do they included here? And how are they ranked? They can rank either based on the gene expression level or based on some other information, how these rows are ranked. And the third dimension is what is the color scale? What is the signal? And what do they mean? So these are the essential elements in such heat map. And these heat map can visualize multiple samples or multiple data sets at the same time to do a comparison. For example, some of you will be reading this paper in this afternoon's discussion session. We can see they are looking at in this plot, they actually align for each region, they align the same regions in these multiple heat maps. Each row represents the same region in the genome, but each each column, they represent different type of signals so that we can compare in a certain region, some signal, you can see some signal from some samples, but do not observe some other signals. And this is another example to compare a transcription factor binding, which is aligned at the center, with a histone modification. So where the histone modification only marked the flanking nucleosomes, flanking these transcription factor binding sites. So another way of further compressing or visualizing these heat map, this signal, is a plot that we usually call this composite plot. The idea is simply to, sorry, this is not aligned very well, but you get the idea. The idea is that you simply just further compress all the rows, the signal from all the rows into one line, into one curve to represent the pattern of these regions. So for example, this one, we observe one peak at the center. And for histone modifications, because they have multiple nucleosomes around, after converting these heat map into a composite plot, we can see this double peak or bunny ears pattern as the histone modification. Question?
Slide.
where it had all the different cancer regions, how, how do you know how they sorted the different cancer regions?
So these regions have to be predefined. You need to know what regions you are looking at. You need to get all the coordinates and to get the signal and pack them together.
Like down the y-axis.
Yeah.
Cancer region, just choose how.
Yes, you can. So in this particular case, so they actually group the enhancer region into 4 different groups. So they, based on their signal, they knew that which signal. So the first group of regions, they only have a strong signal of H3K for monomethylation in these cell type, and they put them here. But within this group, so from this plot, I can tell that within each group, each row, they may just randomly, because we don't see any pattern. Yeah. So they do not have further information about separating different rows. But clearly, they have these 4 different groups in determined before generating these plots. But there are also algorithms, clustering algorithms. So once you get this, based on some signals, they can automatically generate the clustered pattern. But whether the cluster pattern is what you want, we need to think about it to make the decision. Yeah, so you can do it you have the flexibility to do this both ways. So in a supervised way, which means that you give them the information you want, or the unsupervised way. So let some algorithm to automatically determine based on their signal. All right. Yeah, so we're talking about this composite plot. So the composite plot is what are the essential elements? So first, of course, always what type of data, what sample we're looking at, where so data type, we always need to label and put the title and the figure legend. In this example, on the same plot, they are plotting 3 different signals. So the nucleosome-free region and the signal in the nucleosome-free region. I believe this is chromatin accessibility signal, and the blue is nucleosomal signal. It's a blue curve, and the MNA-seq is a different track. And what are the data resources and how they are generating the average signal from all different places in the genome, and always read the x-axis to see what is the anchor, what is the scale. So starting from center at 0 plus or minus 1000, and for y-axis, what are the scale and how the data are normalized. So there are several important points that I want to emphasize when you read or generate a composite plot. So, or several caveats. So first, a peak in a composite plot, no matter whether you see this in this way, or you see this in a very sharp peak, such peak may be contributed by only a tiny fraction of regions. So it does not mean, if you see a peak in a composite plot, it does not necessarily mean that all of the regions you are looking at have such peak. So maybe I can use this histone modification to make it as an example. So we can see the signal strength has a distribution. Some regions have very strong signal, some regions have very weak or even no signal. But if you average them, you can always see the signal. Imagine if we only have a very few regions with very strong signal, and the remaining regions do not have the signal, we can still see the peak in the composite plot. So composite plot only tell you the overall pattern, but it does not guarantee that all of the regions have the same pattern. So they may not be representatives of a global picture. And the other caveat is that a higher peak does not necessarily mean a stronger signal or more region coverage. It highly depends on how you calculate or how you average the signal. Peak is, I mean, the signal is higher depending on the normalization. it may not, it may be because some certain regions have very strong signal, or it may also because of a lot of regions, they all share the similar signal. So usually the composite plot is just for you to visualize whether the signal exists or not, instead of doing a quantitative comparison. So far, so good. Any questions? Good. Let's move on. So Venn diagram is probably we're all familiar with. They can represent our relations between two different or multiple different sets, different sets, to see whether they have shared, overlap, or intersections. So most commonly use the two sets Venn diagram, or we can sometimes use a three sets diagram. But unless absolutely necessary, I do not recommend presenting your data in a diagram that have more than three sets. You can see how complicated such a diagram is, and I think there are some topological problems. So whether it's even mathematically possible to plot high-dimensional or multi-sets diagram, and I saw this is from a paper. It's really difficult to understand, and usually once you want to present some data in a diagram, you want to make the size of the circle and the area of the overlap roughly proportional to the data that you want to represent, right? And this can be easily achieved for two sets Venn diagram, in most cases okay for three sets, but almost impossible for Venn diagram that more than three sets. So try to avoid using Venn diagram to present data that are more than three sets. And so when we compare genomic data sets, sometimes we want to see how many of the genes can have certain signal, or how many of say transcription factors binding sites can have say a histone modification signal. We want to do the comparison or association. The easiest and the most easy to understand way of presenting the data is using a Venn diagram, but in many cases, heat map to do a direct back-to-back comparison, or a scatter plot where we compare the original data points may contain more information than the simple Venn diagram. Because Venn diagram is only a yes or no. You have a cutoff, and it does once you set the cutoff, the other sets maybe just because they have the weaker signal, but some correlation may still exist. So in other words, Venn diagram is very sensitive to the cutoff when you use in the data analysis. So sometimes could give you some misleading results. So once you see a Venn diagram, always pay attention what the data analysis process and whether they could be misleading. So the more informative way is always to read the original or the signal. Another type of plots that we usually see when representing RNA-seq, especially differentially expressed genes from RNA-seq analysis, is called a volcano plot. So the volcano plot is just like a volcano explosion. There are two sides. Usually there's like a valley and two sides. So first it's a scatter plot. So each data point usually represents one gene. And so for RNA-seq, we have the expression information from all the genes in the genome. So there are maybe tens of thousands of data points. But instead of looking at all of the genes, we're only interested in the differentially expressed genes. So how to visualize or represent differentially expressed genes in the volcano plot? Look at those two dimensions. The x-axis usually represents the signal strength. For example, log2 scaled fold change, comparing treatment versus control. And once you give the fold change a log2 conversion, you change the fold change to a number that can be either positive or negative. whereas 1 means twofold, right? Log 2 of 2 is 1, or log 2 of 1/2 is -1. So 1 means twofold, and here we labeled as I think is 1.5-fold. And the y-axis usually represents the statistical significance, and usually represented by the negative log 10 based p-value, or FDR cutoff. So for p-value, it's the statistical assessment of statistical significance of a statistical test. Usually we use the cutoff of 0.05. So smaller than 0.5 means that statistically significance. And in some sense, the p-value or adjusted p-value can represent the level of statistical significance in the data. Although this, if you're interested in more rigorous statistics, this argument may or may not be true, depending on different cases. So after you convert it into a log 10 basis, so I think this is labeled as 1. That means p-value of 0.1. So in this case, the top right corner means the significantly upregulated genes, means that the fold change increase, fold change is greater in treatment versus control. And the blue, the top left corner, means the downregulated genes. So lower expression in treatment compared to control. Okay. So differentially expressed genes can also be represented in different other type of visualization. For example, when we compare the control versus treated, we can use a heat map to represent the expression level of each different gene. So in this case, each row represents 1 gene, and each column is 1 sample. We have 3 replicates for control and 3 replicates for treatment, and the upregulated genes become from blue to red and downregulated from red to blue. Or once we have multiple samples, we want to do a cluster to show the co-expressed pattern of gene sets. We can show it this way, or you want to look at the more detailed gene expression level to label all of this information. Okay, now we'll move on to see how we visualize more complicated IC kind of contact map for 3D genome interaction. So for the earlier lecture this morning, we know Hi-C is a technology to measure the three-dimensional architecture of the genome, or high-order chromatin interactions. So the most common way of visualizing Hi-C or 3D genome interaction is use data representation called contact map. So in a contact map, we can see this is from a famous Hi-C paper 10 years ago. We can see for both X and Y, for both rows and columns, they represent the genomic coordinates, and the color in the heat map represents the contact frequency or the level of signal in each location, means the contact frequency between the 2 locations represented by the column coordinates and the row coordinates respectively. And this plot, it means that the Hi-C data, they can Hi-C can generate 3-dimensional interactions on different length scales, and so they are in very different length scales. So if you zoom in one part, we can see a more detailed pattern of genomic information. And because the entire 3D genome architecture, they are also across multiple different length scales, and they may represent different type of structure, chromosomal structure. So the largest scale is the compartment called A and B compartment, and in the smaller scale, one can observe a topologically associated domains, and the DNA and the most high resolution we can observe DNA loops. Yeah. So what are the essential elements in the Hi-C contact map? just like what I just mentioned. First, the most important thing is scale. So we want to, once we see such a contact map, or some people call this a diamond map, we want to see, we should pay attention that what is the scale that we are looking. So whether we are looking at very large scale compartmental structure or in a very fine scale local DNA interactions. So for example, this plot, they represents 1 MB, 1 megabase, 1 million base pair. So that we're really looking at a very large scale. And this is under a, what is the resolution? So this is under 5 KB resolution, which means each bin is 5 kilobases. And in this case, in this heat map, 5 KB of 1 megabases, so there are 200 bins. 200 times 200 rows and 200 columns. And how they are normalized. We want to pay attention whether the author mentioned whether they normalized these data. And sometimes you want to look for interesting patterns from the high-C maps. So first, blocks. So we can see these type of the whole block. These type of blocks from the data. Or here in this diamond map is perhaps a triangle. So these blocks means that in this certain region in the genome, inside this region, there are a lot of interactions that occur. So any region can have a higher chance to interact with any other region. But these regions have a lower chance to interact with other regions outside this region. So that is represented by this block. So that's inside the block, we have higher signal, and outside the block, we have lower signal. That's how this block occurs. And stripes. So sometimes we can see a line or stripe in the high-C map. That means, what does that mean? So that means that one location in the genome can have a high chance of interacting with many other locations in the genome. So you can imagine this as if there's a loop, but the knot is a loose knot. So one position is fixed and the other position can be sliding across this knot, right? So that's this type of stripe means. So one region can be interacting with many other regions. Question?
Would that be like simultaneous interactions or different interactions?
That's a great question. So keep in mind that usually high-C experiments are generated from a large cell population. So for each cell, it's just a snapshot of whether you capture the interaction at a certain location. So across different cells, the interactions might be captured in different places. And that's how in the high-C data, you observe these type of pattern. But in reality, it just represents an ensemble or average of the interaction pattern across many different cells in the population. But whether each individual cells, this structure can be dynamic. If you believe that these cells are homogeneous, they're from the same cell type, so that you can understand that in the way that this called loop extrusion pattern can occur. But we are definitely not from a high-C experiment. We are definitely not observing the dynamic process of this. So we will just interpret this in this way. Yes.
And just to be sure that in this high-C contact heat map, that diagonal line is just the same. That's the same region of the chromosome, right? And that's why it's very intense because it's the same.
That's exactly right. So we can always see a very strong signal from a diagonal line. And the diagonal line means that the X equals Y. So that's the same region. So always for all of the high-C, for both biological and technical reasons, we can always observe that the most signals occurred in a very local interaction. And so we can also biologically we can imagine that if in the DNA, in the chromatin, the close by regions, they are definitely have more chance to interact with each other compared to very distal, far away regions. But on the other hand, it also represents some technical issues in Hi-C experiments. And that information sometimes we want to be normalized out in the data analysis, because in Hi-C, we're always interested in high water or distal interactions, rather than the interactions on the diagonal.
And where is the stripe in this triangle, Peter? Is it on the base of
The stripe, I mean, is not the diagonal, but it's either on the row or on the column. So in this way of representation, which they switch this to 45 degrees, you might observe a stripe like here or another stripe like here.
But where we see the local interactions, which are diagonal line in the rectangular map, and where is that in the triangle?
Here the triangle is down here, is the base. And that is, these regions are usually not where we are most interested in. Yes?
The red square equals 37 indicate the maximum amount of interactions observed in the sample set?
Yes, in this case, but we should also look at whether this number is normalized, how they normalize, whether there's involved some normalization process. But that means that in this particular data set, it represents the highest, the red means some number of 37. Yeah. All right, and stripes. And so what is loop? Loop means that you observe a red dot in the contact map. So red dot means that in certain regions, compared to two regions, they have a high chance of interaction. Then in the genome, you can imagine that would form a loop. Two specific locations, they are tightly interacting with each other. All right, or in data analysis, sometimes we can consider this loop or this red dot as a two-dimensional peaks, because we're looking at the two-dimensional space when we observe a peak there. All right, I'll briefly mention the circus plot. So they can, this type of plot, they can integrate multiple type of genomic data together, and they can be generated using some software or circuit plot. But unless we really have these many different type of data and we compare to different samples across all different chromosomes, usually this type of data representation, they contain too much information for the readers to understand. So unless you really want to give a huge big picture of a large amount of omics data you generated, I usually do not recommend using a too complicated way of visualizing or presenting the data unless absolutely necessary. So nowadays, single cell genomic technology has been increasingly used to study or to represent the cellular heterogeneity across some tissues or some samples. And the most commonly used way of visualizing single cell data is using a two-dimensional map called a t-SNE or UMAP. So both t-SNE and UMAP are a non-linear dimensionality reduction methods that reduce the entire high-dimensional single cell data into this two-dimensional map. So usually on this UMAP or t-SNE maps, we can observe clustered patterns. So here, each dot represents one cell. And for single cell data, usually one data set can contain at least 100, so usually thousands, if not more than 10,000 cells. And so each data point represents one cell. And the X and Y axis is just the two coordinates that generated in the t-SNE or UMAP dimensionality reduction algorithm. It doesn't really mean anything. It's just the two dimensions. And what we want to observe is the location of different cells. Now, some cells, they located as a cluster in the similar places. And they are far away from other groups of cells. And then one can label the cells based on their samples or based on their any type of information. So this is from the same heat map, but labeled in two different type of labels. So one label is we label them based on their the sample. They have 3 replicates. It means that the 3 replicates, in most cases, the 3 replicates they are evenly distributed in different places. Means that these three samples may not contain very heavy batch effects, or they do not show very big differences across different replicates, maybe unless these class of cells. On the right hand side, I colored the cells based on their classroom pattern. So here, there is a very important point that I want everyone to keep in mind and to pay attention when you read the single cell heat map. I mean single cell UMAP, is that clustering results represented by different colors are different from generating the heat map or a UMAP visualization. So heat map, so clustering is clustering whether the different cells has the same color or different color. This information is calculated from the original data, high dimensional original data. These clustering are not and should never come from this UMAP itself. So a common mistake that a lot of people are doing is that once we generate a UMAP, we can see these cells are together and these cells are not together, and we do a clustering on this 2-dimensional map. And that is a mistake. That is wrong. Because once there's a long linear dimensionality reduction, we lose a lot of information from the original data. And the close by or far away information represent on this t-SNE or UMAP visualization may or may not represent their real relation across different cells. So clustering and visualization should always be done independently, in parallel, not one after the other. Any questions? Here? Yeah.
So this one is the clustering or not?
So the location of each cell comes from a UMAP representation. But the color comes from a separate process of clustering. And that is why in some cases you can observe that say this color give this color, and so on the UMAP, although they are together, but they are not necessarily sharing the same color, because the color clustering information is from a different set of data. Not different, but a original data set. Yes.
So I've been kind of broadly told that the difference between like t-SNE and UMAP projections are just the different like axes, but like I've worked with single cell data sets before, and I know that like the 2 different maps can kind of project cells in a different way. Is there like more detail you can provide on like why they're similar but
Yeah, they are just using different. Well, I won't go to the details of the algorithm, but they are just using different algorithms to do the dimensionality reduction. So you can choose, you can pick either one. It's just for data visualization, and whichever one you think that can better visualize your data, you can pick that one. And always keep in mind that this dimensionality reduction is just for visualization. It's not a process of analyzing the data. So the on the UMAP, on the t-SNE map, if 2 cells are close by, it does not necessarily mean that the actual 2 cells are very similar. Yes.
So then how do you know which analysis method to trust? Because, for example, you see, well, this is a pretty homogeneous data set, but if, for example, you see a blue dot all the way over into one cluster, you see one in the zero cluster, how do you know what exactly, how to categorize that cell?
Right. Always trust your clustering results because that is from the actual data analysis. And always keep in mind that t-SNE or UMAP is only for visualization. Only for visualization. And earlier this year, or late last year, there is a paper published in Nature Communications that presents an algorithm to determine whether the UMAP or t-SNE representation can generate some wrong representation, wrong embedding for different cells. So using that algorithm, one can see that there are some cells they may because they want to keep the global pattern, and some cells they may be distracted to go to the wrong places that they shouldn't be. So again, t-SNE and UMAP is just for global visualization. It's not for you to pick on different cells. And whether the cells belong to the same cluster or not cluster, or not always use a separate algorithm to do the clustering. Yes.
Different sets of 3 and 8 different cells, or are they related? I kind of problem. You have 2 graphs over there. Are they related to each other?
Oh, they are this is the same data sets, the same samples, exactly the same data. So every dot you can find the same cell on another plot in the same location, another cell. But I just use a different color to represent whether this. So on the left, it means that each different dots are from each of the 3 replicates. But on the right, I colored each cell based on the clustering results. Does that answer your question? Yeah, these are the same data sets. So for each cell, they have different assignment, different representations for whether these cells come from which replicates or whether these cells is assigned or categorized into different clusters. Yep. Yeah, again, the axis, the short answer is that you don't want to read more into the axis. It doesn't really mean anything. And in many of the current nowadays in the paper, they don't even label the axis, but just show this is t-SNE1, t-SNE2, or UMAP1 or UMAP2. And it's also a way to tell you that the coordinates for the axis don't really mean anything. It's just a way of better visualizing the data. And some extreme scholars in the field even recommend not using this type of visualization at all. But we think that it's still an easier way to visualize the global picture of single cell data. But always keep in mind that do not be misled by the plot. Yes.
Do you have like proteomics data, look at principal component analysis, if you wanted to look at like clustering variation?
Yes. Principal component analysis is a much better way. And that also includes some fundamentals. So t-SNE and UMAP is you can consider that as a high dimensional, not high dimensional, but nonlinear variation of principal component analysis. The idea is both to visualize high dimensional data in a 2-dimensional map. But PCA is clear, it's more interpretable, it's more explainable. So PCA is a better way if one can observe this in a principal component map. So they use t-SNE or UMAP to visualize single cell data just because PCA does not always work very well. Yeah.
I always struggle trying to figure out how to gain insights from these visualizations. So for example, if we see in 5, there are 2 separate clusters, would that potentially indicate that there's a subpopulation within that initial cluster that warrants further investigation?
The the precise answer is not necessarily.
Yeah.
Not necessarily, yeah. You always, if you can certainly make some hypothesis that the cluster 5 may be consist of 2 separate subclusters, but whether that is true or not, you want to take the original data and redo a cluster to represent. In many cases, it might be true, but it's not guaranteed. All right. Well, yeah, briefly touch the single cell data visualization. And briefly mention another way of representing high dimensional data called bubble plots. It's just like an extension of a scatter plot where we represent where each dot, each data point, they can have 2-dimensional X and Y representations. But the bubble plot contains more information. So instead of each data points can have a size of a circle of a bubble. So the size of the bubble can represent another dimension of information. So this is from a review paper many years ago, talking about all different sequencing-based technologies. And I think the size of the bubble means the number of publications that use certain technology. And the x-axis is the development of technology different years. And the y-axis is the number of citations. So clearly, RNA-seq is the most commonly used sequencing-based technique. But there are so many different ways of different sequencing-based technology to study differences. Okay, brief summary. We show this high-dimensional, high-throughput genomics data can be represented or can be visualized by all different ways: genome browser track, composite curves, heat map, different types of heat map, or high-C heat map, or Venn diagram, and bubble plot, circle plot, all of these. All right, so these are different type of way to visualize and represent this data type. So in the next few minutes, I will share with you some tips about how to present your data in a more rigorous, in a clearer way. So first tip I want to share is that for a scatter plot, if you have a huge number of dots, data points in a scatter plot, in many cases you may not be able to see what are the patterns inside this dense clouds because there are too many data points. But one way to do this is that you can use some heat color or color density to represent the local density of the data points. So we can see these two plots, although they are the same, they all come from the same scatter plot, but they may represent different signal, different information. So on the left, most data points, they are all in the bottom left corner. And on the right, we can see maybe 2 different groups of data points. They are located in different other places. So the density of the data points matters and sometimes can give you more information. There are different ways in R to generate this local density on top of a scatter plot. And in some other cases, you can see if a lot of data points are just stacked in a very small color, it might be because of the distribution of the scale of your data. So in this way, sometimes if you do a log transformation, you may be able to better see the structure in the data. So choose the appropriate scale for plotting, or linear scale, or in a logarithm scale. And one of the most commonly used way of presenting the data is using bar chart, right? And can someone tell me whether there's something wrong with these bar chart? Yes. Yeah, that is a very, very good point. And usually in nowadays, if the data point are very few, we usually are expected to just plot the original data points. But if the data points are not many, I mean, a lot, it may not always be easy to plot all the data points, and a bar chart is fine. Someone else?
The y-axis are not labeled.
Right. See, there are no y-axis. So that is sad. So always remember to label your axis. Okay. Always remember to label your axis. Okay, so once we see the label, do you see other problems with the data, with this data representation? So now I show the labels. These are actual plots, actual figures from a cell paper. Yes?
They're starting at, they all have different starting points and should be normalized to 0.
Right. So they are all on different scales. But for bar chart, keep in mind that bar chart should always start from 0, because the height of the bar is a cube. It gives you some information, right? If you do not start from 0, it can be very misleading, right? So in this case, it starts from 1,000, right? The differences are not as, the actual differences are not as big as it looks. So for bar chart, it should always start from 0. And I give you another example, also from an actual paper published in Cell Reports. If you can see this one, the red dot, the red bars, right? You may, if you directly look at the bar chart, you may think that the first bar and the third bar, they have a huge difference. But if you look at the scale on the y-axis, it may just be a twofold difference, right? So that's a misleading thing if you do not start do not plot the bar chart start from 0. And that is exactly why you should always start from 0 to plot a bar chart, right? Because the height actually really means something, and the differences should not be that high, right? Okay. Another thing is that when you generate a scatter plot to well, you want to present some correlation or some association pattern between different data points, and sometimes the data points they're scattered in all different places on the scatter plot. You may see a positive correlation on this scatter plot, right? But the positive correlations are not that clear. And if we group the cells, group the data points into smaller groups, you may be able to see a better correlation. So in this case, a group based on the x-axis of the data point, I think this is group every 100 or every 200 data point to point 1. So group them if needed. But keep in mind that after you group the data points, you can no longer calculate a correlation coefficient. In the original data point, the scatter plot, you can calculate the correlation coefficient, but after group them, correlation coefficient do not make sense anymore. So think about the extreme case. If you group all the data points into 2 groups, and then the result will be 2 points. 2 data points, the correlation coefficient will definitely be 1. So it doesn't make any sense anymore. But another point to make sure, to keep in mind is that the grouping should be fair. So this is also from a Cell paper. Can you see what's the issue of this plot? Someone? Yes? I'm sorry? Oh, yes. The R value is negative, which is true, right? It's a negative correlation.
There's no consistency in terms of the jumps from half like half flat block.
There's no consistency of how they group the data points, right? First point is smaller than 7, and second is 7 to 19, 22, except for the first one. And from this plot, imagine that if you group the cells evenly, like every 20 data points, would you still, if you group the first data points with the second one, would you still be able to see such a negative correlation? It's questionable, right? So the grouping should always be unbiased, should always be fair. So this is actually a data manipulation that should not be even accepted, even in the in the cell paper. Right. Next example. What are they? Do you see any issues of these two beautiful pictures? They're like cells or chromatin structures? Yes? There is no scale. Yeah. There's no scale, right? So we don't know what they represent. After seeing the scale, the left one is actually from a cell image, and the right plot is from a picture of the Max Webb telescope. So the scale is 0.25 light years. And you can see how beautiful some stars are there, how similar they are to the cells. A very huge scale, but always scale matters, right? Whenever you read or you generate the data. And from this plot, you can see scale does matter. If you can recognize the map of these 3 different countries, scale does matter. Okay, so some take-home messages that always read the axis and pay attention to the scale on the plot. And bar chart should always start from 0. And the bar chart should be on the linear scale, because the height of the bar is a Q and the height of the bar, it represents some information for data visualization. And data point density on the scatter plot is important. And finally, you can group the data points if needed, but do it in an unbiased and a fair way. So just some these take-home tips for you to keep in mind. So I will end my lecture by showing this picture I also got from the internet. I hope you to keep in mind that just having the data is not enough, but how to present the data in a fair way and how to interpret the data actually matters. And that is the way to how you convert the data itself to information that you get knowledge or insights and make new discoveries from the data. And of course, you should always pay attention not to over-interpret or over-critic on the data. All right, so I would like to end here always by sharing with you this picture. Although I do not give you any details of how to generating or how to making these plots, but I hope this lecture can help you better analyze, better read the figures, read the data from published papers, and keep in mind that the data presentation and data visualization matters a lot in the biomedical research. All right, thank you very much. I will end. Yes. Hold on. Let me 1 second.